Hello everyone, my name is Yutong and I am from Carnegie Mellon University. Today I'm going to talk about the joint work with Dr. Carl Kingsford on constructing small genome graphs via string compression. In this talk, I'll present a new framework for genome graph construction that optimizes the size of the constructed genome graph. We draw a natural connection between data compression and genome graph construction. We also address the discrepancy between compressed form and genome graph via the source assignment problem. Although we will not have time to cover this problem in detail, in the talk we have described the details of it in the paper. It is a very interesting and important problem to transform between compression and genome graph. Let me first talk about what is a genome graph. A genome graph is a bidirected graph that stores a set of strings such that each string in a string set can be reconstructed by following a path in G, and we call those paths reconstruction paths. In the example below, the blue arrow and orange arrow represents the reconstruction paths for S1 and S2 respectively. You will notice that in the graph, there will be self-loops or cycles, and in this definition, each edge can be used multiple times. Genome graphs are crucial in pan-genomic studies. The following is a typical pipeline for pan-genomic study that starts by collecting thousands of genomes. Next, we want to create a data structure that stores that large amount of data efficiently. Again, this data structure needs to support the efficient downstream analysis, such as read mapping or variant calling. There has been a line of work on pan-genomic studies showing that analyzing a collection of genomes improves the accuracy of read mapping or variant calling, or using genome graphs, we can discover new genomic sequences from minority populations, and we can use genome graphs to improve the accuracy of haplotype inference. And you'll see that a genome graph is really the centerpiece holding the pan-genomic studies together. And among all the aspects of a genome graph, the size is one of the most important one. Recall that genome graph has to store the thousands of genomes effectively. What we want is to transform a set of strings somehow into a data structure. Here it's a graph. And a graph is consisted of three different components: vertices, edges, and the concatenated node labels. Naturally, we want to minimize the size of it. And our second task is to support efficient downstream analysis. And in fact, the efficiency of the analysis is also directly dependent on the size of a genome graph structure. For example, the sequence-to-graph mapping shown by Jing et al. In 2019, the running time of it is directly dependent on the total number of characters in the node labels and the size of the edge set. So really, our objective here is to optimize the size of the graph structure, which is the vertices, edges, and labels. We have our objective over here. However, directly optimizing the objective will result in a very simple genome graph that we actually do not want, which is a graph like this. A graph like this does not contain any information because any string can be reconstructed on this graph if we can use edges multiple times. And the length of the reconstruction paths will just be equal to the length of the input string. This really just defeats the purpose of efficiently store the strings. So we address this problem by proposing an alternative definition of a genome graph, where each edge can be only traversed at most once. This restriction adds a dependency between the number of edges and the length of the reconstruction path. So by optimizing the objective using the new definition of the genome graph, we are optimizing both the graph structure and the reconstruction path. So in a restricted genome graph, there will be parallel edges between nodes, and the reconstruction paths will use distinct sets of edges. As shown here, the black and orange edges correspond to S1 and S2 respectively. When we are optimizing the size of the graph, it is natural to think about the data compression problem. Here we introduce a data compression scheme named External Pointer Macro, or EPM scheme, proposed by Storr in 1982. In the scheme, one of the important concepts is a pointer. A pointer is a pair of integers that indicates a substring in a string that starts at position with length indicated. And the EPM scheme transforms an input string to an EPM compressed form C that consists of a reference string R and a sequence of pointer to R. To illustrate it, suppose we are given an EPM compressed form, we can reconstruct the input string as follows. We parse the sequence of pointers from left to right and substitute each pointer with their corresponding substrings. So for pointer 1, 4, we locate the substring starting at position 1 with length 4, which is TCGA. And for the second pointer, we locate the substring starting at 3 that corresponds to the substring GAT. And for the green pointer, we do the same thing by appending the corresponding substring GA to the original input string. And this is how we reconstruct a input string, and this is how EPM scheme encodes an input string. You may wonder, why do I focus on the EPM scheme? And this is because there exists similarity between EPM and genome graph. If you view genome graph from a different angle, when you concatenate the node labels in the genome graph, you may notice that it can serve as a reference string where the nodes are simply pointers to R and edges are adjacencies between pointers. And we can see the same 3 components in the EPM form as well. There is a direct correspondence between the concatenated node labels and the reference string. And in a sequence of pointers, there are really just adjacencies between pointers that corresponds to node and edges in the genome graph. To reinforce the similarity, I'll introduce an algorithm that transforms from the EPM form to the genome graph. This algorithm takes a reference string and the sequence of pointers as input. It is a 2-parse algorithm through the sequence of pointers T. In the first pass, it creates nodes by cutting the reference string according to the boundaries of the pointers. For the first orange pointer, we locate its boundary at position 1 and position 5. So we cut the reference string before T and after A to separate the TCGA node. Then for the blue pointer, we do the same thing by cutting at position 3 and position 6. And for the green pointer, we cut at the end of the last node to create a node for GA. Now, you may have noticed that there are actually 2 occurrences of the substring GA on the reference string. And you may wonder, what if we cut the reference string according to the boundary that starts at position 3? And the answer is we'll have fewer nodes. And having fewer nodes means we are having a smaller genome graph. However, using different occurrences in a compressed form does not change the size of the compressed string. So the difference in the number of nodes produced by cutting at different positions for the same substring creates a discrepancy between the compressed form and the genome graph. And we call those occurrences sources. So the choice of the source for each pointer actually affects the number of nodes. We address this problem as a source assignment problem, and we show in the paper that by solving the source assignment problem, we are able to further reduce the size of the genome graph. Now back to our algorithm. In the first pass, we have created nodes by cutting the reference string. Then for the second pass, we first connect all the nodes in their reference order to create the reconstruction path for the reference string. Then we walk again from the beginning of the sequence of pointer T. When we are inside a pointer, which means, for example, we're walking from TC to GA, we just add an edge from TC to GA in the reference order. But when we add the end of the orange pointer, we locate the index that is at the start of the blue pointer, and we add an edge connecting the end of the orange pointer to the start of the blue pointer. In this case, coincidentally, GA node serves as both the end of the orange pointer and the start of the blue pointer, so we add an edge or a self loop on GA. The rest of the pointers follows the 2 set of rules, whether it is within a pointer or it is between pointers. So for the adjacency between the blue and the green pointer, we add an edge jumping from T to GA. The running time of the algorithm is linear in the size of the reference string and the input string T. Now we have the algorithm, and what does the algorithm give us? We show in our paper that there is an upper bound on the genome graph size given the optimal EPM form, which means that we can actually guarantee that the size of a genome graph is bounded by a function using the original terms in the EPM compression form. Here n is the number of unique pointers. And we also introduced an algorithm that transforms a genome graph to a compressed form. And the size of the produced compressed form can be also bounded by the size of the optimal genome graph using the original terms in the genome graph formulation. So the bottom line here is that given a space-efficient compressed string, we are guaranteed a space-efficient genome graph. So far with the C2G transformation algorithm, we're able to solve part of the problem from the set of strings to a genome graph. What we are missing is the algorithm to actually transform the input to a compressed string. And this could be any EPM compression algorithm. The EPM minimization problem, however, is NP hard. So in order to create a genome graph efficiently, we need to find an efficient heuristic. Fortunately, there is an intuitive heuristic named RLZ-graph algorithm proposed by Kruppel et al. In 2010. In this algorithm, the reference string is actually part of the input, and the algorithm parses the input string from left to right greedily. The algorithm runs in linear time in the size of the input string, and it actually can achieve good compression ratio on genomic sequences, and the reference string is intuitively the reference genome. And there can be efficient sequence query indices built on top of the RLZ structure. So back to our problem. Now our input becomes a reference string and a set of input strings. We apply RLZ compression to get our sequence of pointers, and then we run the C2G transformation algorithm to get a genome graph. Again, this RLZ algorithm can be any other EPM compression algorithm. We evaluate the performance of RLZ-graph on human chromosome 1 sequences. The dataset we use are the Human Genome Structural Variant Consortium dataset and the Southern Genome Project. The HGSV dataset consists of a comprehensive set of variants, including SNPs and structural variants. We compare the RLZ graph with the color deburn graphs and the variant graph. The color deburn graph is constructed by Bifrost, and the variant graph is constructed by VG Toolkit. From the dataset, we obtain a set of variants and a reference genome, and we use those as the input directly to the VG Toolkit. But for RLZ graph and Bifrost, we want to know how our methods can perform on a set of complete genomic sequences. So we insert the variants into the reference genome to construct 32 genomic sequences, and we use that as the input. This step also removes pre-processed optimized sets of differences between genomes because the variants are actually identified in sort of like a pre-processing step using the reference genome. We show results on 32 samples from the HGSVC dataset. We show two metrics of a genome graph size. The first metric on the left is the total number of characters in the node labels. As you can see, both VG and RLZ graph use a very small number of characters because both of them use the reference string as backbone, and therefore they produce way fewer number of characters than the color deburn graph under different k-mer settings. And on the right, we evaluate the collective space taken by the vertices, edges, and node labels. And we do that by evaluating the size of the file that represents the graph structure. Specifically, we use the GFA format. And you can see RLZ graph and VG perform similarly, and they both produce smaller graphs than the color deburn graph under different k-mer settings. So we show that RLZ graphs can produce small graphs. We also measure the running time of RLZ graph, and this result is on the 100 samples of human chromosome 1 from the Southern Genome Project. You can see from the numbers from the second column that the running time grows linearly in the size of the input. In our implementation, the RLZ graph runs on one thread only, while Bifrost is run on 30 threads. In conclusion, on the theoretical end, we proposed a new framework for genome graph construction that optimizes the genome graph size for better storage and analysis. We connect the concept of genome graph and the concept of data compression. In the process, we also address the discrepancy between compression and genome graph construction via the source assignment problem. We propose a pair of algorithms to transform one from the other, and through the algorithm, we can obtain an upper bound on the size of the genome graph given an optimal EPM compressed form, or the other way around. On the practical end, we show that our compression-based construction algorithms can produce small genome graphs via the proof of concept implementation RLZ-graph. However, the use of RLZ algorithm does not limit the scope of this framework to one algorithm. Instead, it opens up potential to use other compression forms to construct better and more efficient genome graphs. At the end of the talk, I would like to thank my research group, the Kingsford Group, and especially thank my co-author, Dr. Carl Kingsford. I would also like to thank all my funding resources, and thank you all for listening. I'm ready to take questions.